research also at NASA JPL. And starting off with Jessica, I'll do a brief introduction of each speaker. We'll hear from both speakers, and then we'll do a Q&A session at the end. So Jessica is at JPL, a research scientist in the Origins and Habitability Lab. She earned her PhD in chemistry from MIT in 2019 with Dr. Tim Jameson, and a BS in chemistry from University of Chicago in 2014. Her graduate school research focused on organometallic catalysis and pharmaceutical synthesis. At JPL, Jessica focuses on applying synthetic chemistry techniques to address questions about the origins of life and habitability of the early Earth and other worlds. She experimentally explores mineral-organic interactions and reactivity related to prebiotic chemistry on ocean and terrestrial worlds. Jessica has also been involved in the 2019 Caltech JPL Venus Seminar Series, Enceladus Vent Explorer Mission Concept, and the EELS Path to Flight Working Group. Jessica is a founding member of both the Network for Ocean Worlds Early Career Group, Future Leaders of Ocean Worlds, or FLOW, and the VEXAG, or VEXAG, affiliated Venus Early Career Group Oven. So without further ado, Jessica, you can share your screen and take it away.
Okay, well, thank you very much for the kind introduction and the invitation. And let's see if this wants to work. Okay, cool. Can everyone see my screen? Looks good. So I'm very excited to share with you some of the work that I've been doing to constrain prebiotic chemistry for the origin of metabolism. So, okay, let's see if it wants to switch slides. So I'm sure I don't have to tell people in this room that the decadal survey came out, and I was really excited about it because for the first time, really, I think that life, astrobiology, and habitability were included in a very new way. So there's a variety of unanswered questions about how life originated on Earth, and that, of course, precipitates to other worlds. And addressing this is directly in line with what the decadal wants. So I think that's really interesting and exciting for the field. Importantly for me as a chemist, carbon and organic chemistry makes up a huge part of understanding origins of life, as life is made up of carbon. And so one important part of this, an important part of organic reactivity, is understanding how we can actually get these reactions. And for geologically relevant catalysts, we turn to minerals and metals. And so iron is a great source for this, as iron is abundant in a variety of settings on Earth and other worlds. Mars and meteorites is some examples. And even more importantly, many of these very mineral-rich locations, like hydrothermal vents, have been hypothesized as sites for the origin of life. And we know through a variety of work done experimentally and modeled that iron is capable of mediating organic reactivity. And so for my research, what I seek to look at is, can these minerals mediate reactivity relevant to life or the origin of life? And then what minerals are needed for this organic reactivity? So Earth is really overrun with biology. Biology and life has been very successful on Earth. So how do we actually test this? So the way that we do in the Origins and Habitability Lab is we have a abiotic laboratory setting, and in order to replicate early Earth conditions, we use an anaerobic glove box to remove oxygen. We also, as I mentioned, use iron minerals, and with this, we can adjust pH and temperature conditions to really address different microenvironments that we might want to address. So now moving on to metabolism. So metabolism is a series of life-sustaining chemical reactions. It's broken into two steps. Catabolism takes the food you eat and breaks it down into ATP or an energy currency, and then anabolism builds that back up. And while metabolism is a critical part of life, it ends up being very complex. There's enzymes and cofactors that mediate metabolism, and I'll explain what these are in a few slides. And much like the origin of life, the emergence of metabolism is not currently known. there are a variety of theories and a variety of research about this topic. And so we're aware that metabolism was present during in the last universal common ancestor, but its exact emergence and the timeline of that emergence is still not well understood. And so for this project, I'm really going to be talking about a single reaction. However, I just want to say that metabolism, as we know it, is extremely complex. This is one of my favorite figures, viewing metabolism as a metro map. And we imagine that even a proto-metabolic or abiotic, metabolism network could be quite complex. And so the work that we're doing is part of a much larger picture. And so enzymes are important metabolic catalysts. Enzymes are very specialized proteins that catalyze chemical reactions, an example of which is shown here. So by organic molecule standards, these are very large and very specific complex structures. They're often very substrate specific. And we mostly think of bonds being, structural for organic chemistry, but these have much larger, structures like these helixes and sheets that are extremely complex. And the short version of this is that this complexity was likely not available at the origin of life. So we must investigate alternatives for enzymes in these reactions. However, cofactors are non-protein compounds that are required for enzymes to be effective, still very important, but much less complex in the sense of relative to enzymes. And so just in your everyday life, most vitamins act as cofactors. And there are a variety of kinds of cofactors, but today we're only focusing on one kind, which is electron transfer cofactors. All these do is transfer an electron back and forth and, shuttle these electrons, for chemistry to occur. And even within that, I'm going to be talking about two main cofactors, NAD and NADH, and quinone and hydroquinone, which is, as a spoiler for Katherine's really excellent talk that's coming up, is the continuation of this work that I will hint at, but leave most of it to Katherine to discuss. And so here's just an example of a cycle that's believed to be ancient. So this is the citric acid or Krebs cycle, and it's currently used by all aerobic organisms. And its goal is to generate ATP. But you'll notice NAD and NADH make a number of appearances in this cycle, making it a very important and relevant cofactor to look at. And so I'd said earlier that enzymes probably aren't around at the origin of life, so we need some analog to test our reactions with. And for this, we turned to iron sulfide minerals. And we did this because structurally, the iron sulfide minerals are similar to iron sulfur clusters, which are important cofactors found in all living organisms. And with their structural similarity, we wanted to test if they would behave similarly in reactivity. And these iron sulfur clusters are very important for metabolism and play roles in many biological processes and interact with the cofactors that we're interested in. So we thought this was a good place to start. So for our original project goals, we wanted to test the reduction of NAD to NADH under early Earth conditions. And specifically, we were testing a hypothesis by Lascano and Miller that metabolic reactions could be catalyzed by a non-protein catalyst like an iron mineral. And we wanted to do this to see if the iron compounds could facilitate this reaction. And we wanted to do that in order to answer questions about the origin of metabolism on Earth, as well as the origin of metabolism on other worlds. So as I said, we'll be using NAD as our cofactor. NAD for organic molecules is pretty large, all things considered, but you'll see it abbreviated in the slides to come. So NADH, I will abbreviate as this ring up here, and NAD as the, its aromatic component. NAD is found in all living cells and is an important electron transfer cofactor. So before we got into the experiments, we wanted to do some modeling to understand the feasibility of these reactions. And so we collaborated with Doug LaRoe at USC to test a couple different reactions with FES minerals representing different iron sulfide minerals. And we also had looked into dissolved iron too. And so the punchline of this is that many of the reactions seem promising, and they seem promising across a range of temperatures. So as we went into our experimental set, we were very confident that we might get something that worked. And so for our experimental design, inside of our nitrogen-filled glove box, we took NAD, our desired iron source, whether it was a synthetic iron 2 mineral or dissolved Fe2 plus in water, and stirred that for a set amount of time. We had ran these reactions on the scale of about a week, sampling with a pipette at T=0, T=3 days, and T=1 week. And then once we had those samples, we had a number of different things we tested for analysis. So UV-Vis is really commonly used to look at compounds like this, and we found that UV-Vis was the most successful for us. As you can see with this figure here, the iron really does not have a strong signal, but the lightest blue is NAD, which does not have a signal at 340, but NADH has a very clear peak. And so with NAD as our starting material, we could very clearly track transformation to NADH with growth of that peak. We also tried nuclear magnetic resonance, or NMR, but we had challenges with this technique due to the amount of iron in reaction and difficulties purging the iron from our samples. So I want to walk you through the general experiments we ran. These were ran in multiple runs, but for this table, I'm going to lay out the experiment, the NAD concentration, what iron we used, any considerations for conditions, i.e. temperature, stirring, and then the time points. So for the time points, gray blocks means that there was no peak observed at 340, and therefore there was no NADH. So no desired reaction occurred. Green indicates a positive peak at 340 nanometers and detection of NADH. And the star is for inconclusive results. For some of these, we saw tailing, making it not clear whether or not we were generating NADH. So I started at 1 millimolar concentration, which is comparable to how the concentration of NAD within a cell, and with either no iron as our control, the synthetic FES or Fe2 plus or a mix, we saw no reaction at this under these conditions. So with this, we wanted to test a higher concentration. So I moved up to 100 millimolar NAD. And for this, we were starting to see some signs that we might be moving in the right direction. So with our FES heated, we did start to see tailing, and with the Fe2 plus, we also saw tailing. And so I increased the concentration further to 500 millimolar. And while in the control with no iron, we saw no reaction. We were very pleased to see a distinct peak of FES at T=72 hours and T=1 week with FES. So in this, we were able to show, no reaction without the FES, reaction with the FES. With the Fe2 plus, we were still getting tailing. And so at this point, we wanted to test other parameters around the FES successful conditions. And so we tested heating, and this resulted in tailing and possible decomposition. Without stirring, the reaction was not successful. And then when the reaction was exposed to oxygen, we also did not see a clean peak. And for other controls, I also tested elemental sulfur as well as no mineral and open to oxygen. And for this, we also saw no reaction. So in summary, NADH was definitively observed with 500 millimolar NAD and FES, and the NADH was only observed with the FES. And so here's the spectra of the reaction. And so you can see our controls as well as the reaction, which has a very clean peak at 340 nanometers. This reaction was very sensitive to mixing temperature and concentration. And in some cases, as I noted previously, we did get this tailing. And so for the tailing, here are two examples of what that looks like. So there is an increased absorbance at the desired where the peak would be, but there was no clean peak that we could integrate and get yields. And so interestingly, the tailing only occurred in cases with iron. And so we do still think iron is critical to our reaction. And so I said earlier in the talk that the model was very positive. It looked like we were going to get good results. Everyone was very excited. And we only had one successful result. And even then, the yields were fairly low for this reaction. And we do believe that this is due to stability of NAD and NADH, which are both not stable compounds, but NADH was observed to be especially unstable. And so we believe that we are decomposing our product during the reaction, and we could be, generating and then decomposing it. Before I had started running the reactions, I ran a number of controls to see how stable these reagents were to different conditions. And NADH was found to not be stable to heat. Interestingly, it mostly turned back into NAD, and it was especially sensitive to pH changes. And the results of this study, we think, are positive and do indicate that this is a possible reaction. However, we think these would be very challenging molecules to study, for spaceflight, or, with something that, has them sitting out for a long period of time. And so one last test we wanted to run was to understand the structure of NADH. It has a number of isomers, and only one is used by life, and that's the 1,4 isomer. And so we use this enzyme test where basically you added the LDH enzyme along with pyruvate, and it should convert your NADH, if it's the proper form that life uses, back into NAD. And so the way you do this is you would take your reaction, you would take something you've already analyzed on the UV vis, and then you would add this mixture in, and you would see if the peak got lower over time. And the results of this were inconclusive. We got signal overlap, and we do hypothesize, based on previous work synthesizing these molecules, that it is primarily the 1,6 form. However, as we're looking at this being an origin of metabolism well before life, we're not particularly concerned with the form of the structure of the product. We're mostly concerned whether or not it's redox active and can continue in these cycles. And both of these forms should have no issue being redox active. And so overall, we've shown that a simple FES mineral can mediate the reduction of NAD to NADH, which is an important metabolic redox reaction. Metabolism could have initially been mineral driven, possibly based on these results. And if this is the case, worlds that host mineral water interactions could host these reactions. That all being said, it's very exciting, but this is really one reaction, and metabolism is very complex. So what other metabolic reactions could behave similarly? And so Katherine's going to give you a really, really excellent talk in a few minutes on what our next steps were. But the short version is we turned to the electron transport chain and wanted to see what else we could pick. And, some broad takeaways about this work are that these laboratory studies are directly relevant to the planetary science and astrobiology decadal survey. Abiotic chemistry is capable of being extremely complex, even without life. These cofactor reactions can occur abiotically, which addresses the origin of metabolism. And these laboratory analogs are unique settings where we can actually explore abiotic chemistry given Earth's biosphere. And so with that, I'd like to thank my really fantastic co-authors on this paper. Lori Barge and Scott Pearl are co-PIs of the lab that I'm a scientist in, the OHL, and we had a variety of excellent collaborators. And we have a number of students and postdocs, and you should feel free to reach out if you're interested in that as well. Thank you very much for your time, and I'll be happy to take any questions after Katherine's talk.
Okay, so it seems that one of our hosts, Becca, had some problems. So I think I can introduce Katherine herself. So let me pull up that bio really quick. Yes, so our next speaker is going to be Katherine Dzurilla, who is a postdoc at JPL working in origins and habitability lab. And after graduating from Oklahoma State University with majors in biochemistry and physics, she completed her PhD at the University of Arkansas and her PhD thesis focused on the solubility of tholins within Titan lakes and their potential to form biologically relevant compounds. And during her graduate work, she became interested in investigating early biological systems and how these compounds might exist. And then she pursued a postdoc position that incorporated the origin of life studies. And now her current work is focusing on ocean worlds and their potential to hold biochemical reactions. And so thank you.
All right, hello everyone. Is my screen sharing? Should be. No. Is it working? Oh, okay. Awesome. All right. Thank you for the introduction. I'm really excited to talk to you guys about, as Jessica mentioned, a study that came out of. Oh. Okay. I'm really excited to talk to you guys about one of these studies that came out of the investigations that Jessica was talking about biological compounds and the interaction with minerals. So I'm really excited to present to you guys today the potential for abiotic reduction of quinones through mineral interactions. So as Jessica was mentioning, metabolisms, as we understand them today in cells, are extremely complex. They involve a lot of different reactions that are involving a lot of different proteins and cofactors that are all shared in between different cellular functions and biochemical reactions. As she mentioned, one of those is specifically within the electron transport chain, the NADH to NAD reaction that can be facilitated with the iron sulfur clusters. However, even in the electron transport chain, that's not going to be the only reaction that requires some type of mineral interaction, specifically the FES clusters. Jessica talked about one of the first reactions in the electron transport chain, the NADH to NAD reaction. My project is investigating one of the next reactions that's facilitating this proton movement through the electron transport chain. We are looking at ubiquinone, or you might also know this enzyme as coenzyme Q10, and we're looking at its reactions in the electron transport chain. If we're going to zoom out and we're going to think about the electron transport chain in its totality and think about what its fundamental function is, its ultimate goal is to produce ATP from ADP, to add another phosphorus onto the original ATP. And the way it does that is through a series of complexes in the bilipid layer of the mitochondria. The electrons will be brought into complex 1 and then be brought all the way through to complex 4, and then eventually the ADP will be brought to ATP through the ATP synthase. However, these reactions are not just occurring in the complexes. It's all going to be a system that involves different proteins and cofactors. And ubiquinone is one of those cofactors that is really important in the transfer of protons through the electron transport chain and through all of these complexes. So what ubiquinone does is it specifically picks up electrons from complex 1 and 2 and then transports them all the way over to complex 3. And the way it does this is through a series of reductive reactions. It's actually going to be physically picking up that hydrogen from complex 1 and 2, moving it through the bilipid layer, and then dropping them off at complex 3. And if we look at the structure of ubiquinone, we can see there are a lot of different things going on. It has that aromatic or that ring structure in the middle. It has that different groups that are sticking out the sides, and it even has that really long carbon chain over on its side. However, if we were to break this down and to think about its function, what is the goal in the electron transport chain? The way that it brings the hydrogens all the way through to the different complexes is by losing one of the double bonds for the oxygen and then picking up a hydrogen as it moves through the different complexes. So that double bonded O becomes a single bonded hydroxyl group. If we were going to break this down and think about its reactions in its simplest form, we would really want to focus on that specific reaction where it's picking up that hydrogen and moving through the electron transport chain, as that is going to be what is essential for the electron transport chain to function. So if we were going to break off all of the other things, this ring structure in the middle, this quinone structure with the ring in the middle and the 2 oxygens on either side, that is going to be the most important part of this structure. That is going to be what is actually facilitating the biochemical reactions that are so fundamental for the electron transport chain. And what's interesting is that if we look specifically at quinone structures as a whole, previous studies have been able to show that quinone structures can undergo these redox reactions abiotically, specifically with iron. And this is really relevant to a lot of early Earth research, as iron, as Jessica mentioned, was abundant during the early Earth, and is thought to be present in a lot of areas that we think that life emerged from. So understanding how quinones react in these environments could provide a lot of information to the origins of the electron transport chain. So I know Jessica went over this, so I will briefly do a quick refresher, but the iron-sulfur clusters are really important cofactors in the electron transport chain today. Specifically, previous work has been able to show that prebiotic or abiotic reactions that are similar to reactions we see on life now have been able to be facilitated through iron-sulfur clusters or iron-sulfur minerals. What's also interesting is these were abundant in hydrothermal vent areas, again, leading us who study early Earth to really look at it as one of the minerals that could have played a large role in life's evolution and progression. So the iron sulfur would be most likely be present in these early Earth environments, but that's only one part of the reaction. We also need these quinone structures to be present in order for this full reaction to occur. So the next question becomes, were quinones even present in these environments? And the answer is quite possibly. Previous work has been able to show that quinone structures can be produced through interactions of different minerals and polyaromatic hydrocarbons. Additionally, some recent work was actually able to show that quinones within meteorites were able to undergo redox reactions in different solvents. So while not definitive, the probability of quinone structures being present in these interactions is not zero. They definitely could have been there. And if so, they could have been some of the earliest forms of these reactions that are so integral to biological functions, such as the electron transport chain today. And that's really what this research is getting at. We are wanting to observe what would these very early reactions look like? Could they even occur within these environments? So what we did is we decided to really break down ubiquinone into its simplest form, the most important part that we see in the electron transport chain today, that quinone structure. We selected benzoquinone with the two oxygens on either side, and we asked the question of whether benzoquinone could be reduced to hydroquinone through interactions of FES. And because we really wanted to investigate, the origins of the electron transport chain, we ran this under early Earth conditions to see could this reaction even occur. So to do this, like I mentioned before, we selected benzoquinone and we added it to some milk water. And we did this in addition to iron or FES. We did this in multiple different equivalencies. So we added an increase in volume of FES in 1 equivalency, 5 equivalencies, and 10 equivalencies. We did use some other minerals. We did use FeCl2. However, spoiler alert, we really didn't see anything with that mineral. However, we did think it was important to continuously test other types of minerals that might be present. Because we were wanting to observe this in early Earth environments, like Jessica mentioned, we do have the ability to do that in the OHL. So these reactants were allowed to react in Arcoy and in anoxic conditions, and they were allowed to react at room temperature for different hours, and we sampled them at each time point. We sampled these reactions at 0 hours, meaning right after the FES was added, and then at 24 hours, 1 week, 2 weeks, 3 weeks, and 4 weeks. All of these aliquots were taken and then analyzed with nuclear magnetic resonance. And we used NMR to really observe the structures and if the benzoquinone compound would be able to be reduced in this environment. And we selected NMR for a couple different reasons. However, one of them is that it is a really straightforward way to observe the shift from benzoquinone to hydroquinone. So the typical spectra for benzoquinone is going to show 2 peaks. One is going to be around 6.8, and the other one is going to be around 6.79. There's going to be some ranges there, but in general, benzoquinone is going to have those 2 peaks that are close to each other. However, a solution that only has hydroquinone in it is going to have 1 peak, one singular peak that is at a 6.79. And so we used mostly proton NMR to kind of gauge what compounds are present in each of the samples. However, we also did use carbon NMR on some of it because we wanted to be sure on the structures that were being produced. Again, we're really looking at the oxygens that were on these quinone structures. And we just wanted to make sure that the structures that would be produced in these solutions were indeed the reduced form of benzoquinone, which would be hydroquinone. In a control, like I mentioned before, with just benzoquinone and water, we were able to see the benzoquinone peak and the hydroquinone peak in just a typical solution. And that's to be expected with compounds of benzoquinone. A little bit of hydroquinone is going to be present. And so this is very typical for a sample that has just benzoquinone in it. What's interesting, though, is that when we add FES to these solutions, we actually do see some changes, especially in the peaks. So as you can see here, and with equivalency of 1 out of a sample taken at 1 week, we do see 2 peaks. The benzoquinone is definitely present. However, as we increase the equivalencies at 1 week, we do see the disappearance of that benzoquinone peak and leaving us with just the hydroquinone peak, meaning that almost all of the benzoquinone that we can detect has been reduced all the way to hydroquinone. And what's interesting is that this was the speed of this reaction was dependent on the equivalency. So it was dependent on how much FES we added to the solutions. To kind of break this all down and show you what we saw, we were able to run these at, like I said, 3 different equivalencies, 1, 5, and 10. And at each of those equivalencies, at some point, we were able to see a full reduction of benzoquinone, meaning we did not see the benzoquinone peak anymore. We only saw that singular hydroquinone peak. And for equivalencies 1 and 5, that was able to be seen around 4 weeks. However, when we increased that to 10 equivalencies of FES, we were able to see that complete reduction or that singular peak of hydroquinone as early as 1 week. Like I mentioned before, we did test some ferrous chloride. However, there was really no reaction. We just saw the benzoquinone and the hydroquinone, the 2 peaks within all the equivalencies at all time points. So what can this tell us? Well, without FES, the benzoquinone is largely going to stay oxidized. It's not going to fully reduce to that singular hydroquinone peak. There's always going to be some benzoquinone present. And additionally, it is the FES that was important in this reaction. The ferrous chloride did not yield the same type of results as the FES. And what's also important to note from these results is the reactions are going to occur more quickly as we increase the FES concentration or the volume. The more FES we put in there so far that we've seen in our results, the faster that the benzoquinone was able to be reduced to hydroquinone. This can tell us a lot about potential reactions occurring on the early Earth. If the benzoquinones were present and they interacted with any of the FES that was in these environments, it is quite possible that they would undergo these same reduction reactions that we see in the electron transport chain today. And this actually might be some of the earliest forms of these reactions that might have been built upon as time went on, leading to the cellular respiration as we understand it. Especially, as Jessica mentioned, in these hydrothermal vent areas, any FES present, if it did react with these quinones, could be incorporated into larger chemical systems. So future directions. Obviously, FES is great. It has been able to be, it's seen in cellular respiration today. It's also been shown to abiotically reproduce the same reactions we see in biology today. However, we all are wanting to test other minerals to see if we're able to replicate those reactions with something else. So we have been talking about adding NaS or just even elemental sulfur to see if those minerals can produce the same results. Additionally, we're wanting to see if this can occur across membranes. For the electron transport chain, membrane separation is super important to facilitate that electron and proton gradient that is necessary for the entire process to occur. So seeing if this reduction can happen across membranes would be really interesting. Additionally, keen viewers might have also picked up that benzoquinone is going to be a lot more soluble in water than hydroquinone. So doing some experiments in mixed phase investigations might provide a little bit more information on how this reaction occurs and what environments it can occur in best. Additionally, we're wanting to look at the mineral precipitates that we're getting from these samples. There's always something left in the bottom of the vacuum tube, so looking at if there are any changes there can tell us more about the reactions, what environments they can occur in. And that's really important when we are looking not just for how life evolved here on Earth, but also looking for life on other planets. In places like Enceladus, where hydrothermal vents are thought to occur, knowing how different minerals can interact with any organics there can inform our understanding of how we think about not only how life originated on Earth, but also how we should be looking for life on other planets. I will also acknowledge the OHL, specifically Dr. Jessica Weber, for really spearheading this and leading the project, and all the other people in the OHL. This project could not have happened with you, and we are all so grateful. So in conclusion, FES has been observed in multiple different laboratory studies to facilitate reactions that are really common to really similar to the reactions we see in our bodies today. Specifically, NAD to NADH has been able to be replicated in laboratory settings, as well as the production of a simplified version of ubiquinone. Also, replicating the biological reactions in abiotic conditions can really inform how life could have originated and also kind of happened on early Earth. And we can take that information into how we think about other planetary bodies and whether prebiotic reactions could occur in that environment. So with that, I will leave it on my acknowledgements, and I'm really excited to take any questions. Thank you.
Thank you so much, Katherine. And also thank you, Jessica, for your great talks today, shedding light on some of these early possible reactions. So we will jump into a Q&A session now, and you all should be able to put questions in the chat. And in that case, I can read them so we can all think about them together. And also you can raise your hand so we can unmute you and you can ask your question directly. We're a little ahead of schedule, so usually we go till 1/2 the hour, or we could just go till we get through all your questions, whichever's sooner. All right, so I see that Kevin has his hand raised, so let's go ahead and get you unmuted, Kevin.
Okay, can you hear me okay?
Yeah.
Okay, thanks. Two fascinating talks. It's nice to see organic chemistry at the heart of prebiotic chemical research. So, got a question mainly for Jessica. Very interesting that you're seeing a clear generation of NADH, and it's extremely unstable in aqueous solution because it's a good hydride donor. My question is, if you were to add a certain concentration of long chain fatty acids to form vesicle, if you were able to sort of encapsulate your NADH inside vesicles, this might change things, because then you could get maybe, I'm almost certain you could get some of the iron sulfide diffusing into there, and then you'd have a more controlled cell type environment in which you could actually maybe see an increased concentration of NADH and also carry out some fundamental bioreduction reactions like you showed with pyruvate. So have you thought about doing something like that?
Yes, so there is, so this is unfortunately going to be a longer answer, so I do apologize. There is some really excellent work looking at reduction or oxidation of these compounds with inside of like vesicles. I believe like Nita Sahai has a paper out about this, and there are a few others. We specifically did not want to do that in the first round. More so looking at, something that was really prebiotic. We did not, there was no buffer. We did not modulate the pH. We did not, really, it was just iron compound and water. And we thought that was a very critical step in understanding this. There are a number of directions we can go. I do think that looking at, and actually I'm going to answer a question in the chat too, because pH was not controlled specifically to be a prebiotic environment. So I do think that a good next step would be looking at something like that to do that. but I think that there is some precedent for,
and I could imagine if you do a set of control pH conditions, you'll see, I would predict you'd see much less NADH generation at lower pH than you would at, say, between pHs 7 to 9 would be probably the optimal. I predict that's when you'll see your maximum.
So we started at our goal was to start at about pH like 6.5, between 6 and 7, because we found that both NAD and NADH are unstable to large pH changes in basic conditions like the ribose linkage cleaves, and bad things happen in acid is the short version.
Oh yeah, absolutely. And especially when you've got a hydride donor in acid, as we know, protons can be solubilized, but hydride ions cannot. And despite the fact biology makes use of hydride transfer reactions to carry out reductions and hydride removals to carry out oxidations, which is fascinating, but it does so in a very controlled enzyme sort of complex, which is elimination of solvent water, So that's why I think vesicles could be an answer to taking this forward, because you can then possibly concentrate it down, have far less water present, and create a sort of abiotic cellular type environment, I think.
Yeah, I think that's a really good idea. And I think that, this was very much a pilot study, and there's really a lot more that I'd like to do with it.
No, it's very, very good. I mean, I think you got some very interesting results, I have to say. It's very good. First time I've seen actually looking at NADH, and it's interesting. It's iron sulfide, which is what does it. No other iron compounds. Iron oxides, I'm pretty sure they're not particularly soluble. For one thing, iron chloride you showed does not work. So it's iron sulfide is key, and that's why it is present to this day within ourselves and every other living organism on this planet, and almost certainly was in the last universal common ancestor. Well, thank you. It was really good, and thank you, Katherine, as well. With that was very interesting indeed. The quinones. I mean, as I say, just a quick question for Katherine, if I can, about the origins of quinones. You say they're found in meteorites. I mean, predominantly in the oxidized form, I presume, or the hydroform. What was the ratio? What's the ratio? I've not seen, because I've seen lots of things on things like amino acids, carboxylic acids in meteorites, but not quinones and hydroquinones. What's the approximate ratio of quinones to hydroquinones within meteorites? Question for Katherine.
Yeah, that's a really good question. I, like you, when reading about this, mostly focused on like the amino acids, and so I also was really surprised about the ratio. I haven't really come across anything to quantify it, but to be fair, there's probably some stuff out there that I have missed. But yeah, it's an interesting thing that I don't think a lot of people have been looking at, because the ratio would be really important when you're looking at these reactions.
Okay, thank you, Katherine. Thank you, Jessica. Two fascinating talks. Thank you.
And thank you for your questions, Kevin. I see some questions also in the chat. We have one from Willie Muller, who is asking of Jessica whether you could reproduce the single positive results. So in other words, is it possible that this was a randomly occurring event? We don't understand yet. And we don't understand yet what conditions make it possible. So we can start there, and then there's a second question that Julie has.
Yes, well. So I didn't get into this due to time, but we ran these in triplicate, and so that's why the yields had an average plus or minus on it. So this was a replicatable result.
Great. And then the second question he has is, since the NADH seems to be unstable at several of the conditions you studied, could it be that several other of the conditions are generating NADH but are leading to short NADH lifetimes?
Yes, so we do think, for especially at the number of lower concentrations, we are butting up against our detection limits, and especially in the tailing, that we certainly don't have a good feel for what is underneath that line. We do think that there's probably decomposition, but it doesn't rule out the presence of NADH.
Thank you. All right, next question from Sarah Smith is, we addressed the pH not being controlled. Okay, so I think we answered that question. So there's a second part of the question, also wondering if water activity was measured during any experiments.
We haven't looked into that. That is a really interesting parameter. But like I said, this is the NAD study is a pretty like preliminary proof of concept that we hope to expand on.
Okay, so here's one from Donald Burke for both speakers. He says, to observe a reaction, you need both a thermodynamic drive and kinetic pathway. A key hallmark of biology is to accelerate kinetics. What would be the most successful / promising / realistic catalysis, mineral or organic, to add to your respective systems?
Katherine, do you want to take a swing at that, or do you want me to go ahead? Okay, I have a lot of thoughts. So we are looking at this point at very specific, discrete kind of reactions. However, I think in the end, what I'd really like to be able to do is dump everything together, shake it up, and have some kind of cycle, lots of beautiful catalysis. And I think that in some of the other work, it's the photochemistry is used to drive the reaction. And I think actually there's also really excellent work using some kind of metal peptide complex to drive the reaction. And so I think that, really to go in, not to answer 2 questions at once, but to really go into looking at, my first step would be looking at other minerals. We used one mineral, and I cut this from the slides because it was very, it's not that exciting, but we wanted to use a synthetic mineral that we could control and know, life had not climbed all over. But I think initially looking at other minerals to start and mineral mixes is a good idea there. And then in the reduction, a lot of times when I've talked about this, I've been asked like where the hydrogen, where is this coming from? Is it something that's happening with water? And so I think that for me, for this question, really probing that is a key part of this. And maybe that is a non-answer on what exactly I'd add, but I think those are the key next steps that I would go to narrow that down.
I'll just check back with you, Katherine, if you had ideas of things that you would add to potentially enhance the catalysis in your reactions.
I think Jessica covered it.
Great, yeah. Yeah, I also like the idea of the addition of peptides or even amino acids. That might be interesting. All right, so we have a question from Sarah Smith. Will you attempt to use a range of different iron sulfide minerals in future experiments? So I think we kind of touched on that, but maybe if you could expand on some of what those iron sulfide minerals might be, or other minerals.
At the beginning of this project, I really wanted to use greigite, which is not the most stable of minerals, but it has a good structure that we were looking at, and we were really hoping to be able to synthesize it ourselves in good batches. But I think that for adding the iron sulfide minerals, we really need to be careful about not just making sure they're clean, but making sure we're consistent. So, one of the early questions, which is excellent, is did you repeat this reaction, which is really critical to understanding if this is, a standard thing that occurs. And I think that it's really hard to do replicates if you have variability in your mineral. And so I think that that's something that Katherine and I have been talking about, and we are, looking for other minerals, but we want to make sure as we approach this, we're doing it in a very rigorous way.
Yeah, that makes a lot of sense. All right, so next question we have in the chat is from Juliana Di Giacomo, who compliments you on your presentation and has a question for Jessica. So when metalloproteins, which catalyze metabolism, first emerged, do you believe that they would have incorporated iron sulfides or other forms of iron containing clusters, like 2 iron, 2 sulfur, etc.?
That is a really excellent question, and I'm not sure. I think that there is work being done on some of that, but there's a lot of theories, and I really don't think we know.
Fair enough, fair enough. All right. So that's the end of our current list of questions in the chat, but just a reminder, you all can continue to add there and raise your hand if anything comes to mind. I, in the meantime, have a question, which is, so the scenario you're imagining involves hydrothermal vents. I don't know a lot about them, but I'm assuming they have variable temperatures. So my question is whether you've looked into the effect of different temperatures and whether that would influence the balance of the reduced versus the oxidized state in your reactions.
Yes, it definitely does. And I should say, I'm by no means advocating that this is occurring, perfectly the same way under hydrothermal settings. There's a lot of places where iron sulfide minerals are. It does make for very cool pictures to include hydrothermal vents, though, in my opinion. But I think that in any experimental setup, you have to make assumptions. And the fact is, it's much easier to run something at standard temperature and pressure than to pressurize it and heat it. But I think that that would be something I'd like to test. And we also found more decomposition at 70 degrees C, although that maybe we really don't know what's happening in the reaction. It could be generating faster and then immediately decomposing. And so my thought on that, selfishly, I'd really like it to, if we could add something else to trap it, maybe it would still work. And so I think that we certainly have more early Earth conditions to test, but that's where it stands on the NAD. So Katherine, I don't know if you want to add about the quinones.
Yeah, I mean, for the quinone project, we also did do some temperature dependent studies, and with the results that we were seeing, the reduction actually occurred much more quickly. Regardless of the equivalencies, we were seeing reduction at like as early as 1 week.
All right. So while we're waiting for maybe any last questions coming in, I'll remind people that we do this every 3 weeks. So be sure to join us again. We'll have a new topic and speakers for you. And then we do have a Slack group that you can participate in. I'll pop a link for that in the chat momentarily. But I really just want to thank our speakers again for their great talks and their great research that they're doing and sharing it with us, and everyone for coming. We really appreciate it. None of this would happen if we didn't have an audience. So again, thanks for coming. I'll put the Slack information in the chat, and we'll see you all next time.
Thank you, Jessica and Katherine. It's too difficult to imagine the hydrothermal vents, the pressure, the temperature. It's about sometimes 400 degrees and the pressure incredible. Yeah. Thank you.
I agree. Great experiments. Thank you so much.